Hello, hello. Good day, everyone. Welcome to Chapter 9 on the respiratory system. Now, when we think of the respiratory system, many of us think about items like oxygen concentration, carbon dioxide concentration, our lungs, our trachea, maybe even our alveoli, and you're all correct in that. So what this chapter is going to explore is how exactly different animal models will utilize gas exchange with the environment. And we can have many different strategies to do this, anything from as simple as just doing a diffusion mechanism where the air is moved from a high to a low concentration, thereby also moving their gases like oxygen and carbon dioxide as needed, to when items get a little bit larger or organisms get a bit larger, that they're able to generate bulk flow through muscle contraction, or even the appearance of specialized organs and structures like lungs and gills. So on the illustration that we see on this particular slide, it's just a brief little overview, and it's going to be some of the items that we're going to talk about as we move through the chapter. The fact that if you're going to be utilizing your respiratory system, that your ultimate goal is often to go ahead and have some type of gas exchange between the environment and respiratory surfaces or within the body. So we can start off rather small, and what we see happening is that in very simplistic organisms, a lot of times they'll just be able to get all that done with diffusion, which is basically moving the oxygen from a high concentration through a low concentration, allowing it to migrate from external to internal. This, of course, can be done both in a water as well as in an air type environment. When we take a look at organisms that, such for instance, like the Cnidarians with the sponges and the hydras, what we see happening is that those organisms will tend to go ahead and have contractions, or they'll utilize bulk flow. Bulk flow will allow the water, because if they're in an aquatic environment, to move through the external compartments in the body to enter inside into a cavity. And as the water is entering into the cavity, it often interacts with the cells that line that cavity so that the cells can extract the oxygen and any additional gases or nutrients that they might require from the water before it's exited back out of the organism's body. You can also see that if you concentrate on the right-hand side of the illustration, when you take a look at samples like leeches and earthworms, these worms can use diffusion and gas exchange and directly communicate with their external environment. And part of that is because the skin tends to be relatively thin, so we can go ahead and utilize that to directly move the gases from one side to the next. In some animal models, we're going to go ahead and come across some specialized structures. So for instance, when we take a look at our insects, we're going to talk about the fact that they have a tracheal system that's set up that will work exclusively with flowing the air also in bulk flow. And as it's doing so, it's going to flow through specialized items called tracheas, and that's going to allow them to directly interact the gas with the hemolymph that's circulating throughout their body. When we take a look at vertebrates, mammals, and whatnot, what we see happening is that we will have the highest degree of specialization when it comes to organs, and that will have a play with the fact that we'll have multi-lobe lungs that will be dedicated, that will be filled with alveoli that will do nothing but gas exchange, allowing oxygen to enter and carbon dioxide to exit the body. So as you can see right here, taking the concept of the chart and just kind of put it in written format, it says that if you're looking at animals that are very simplistic, they can get away with doing simple diffusion. However, when they start getting increasing in size, we have to take a look at what type of methods they're going to be utilizing. So you can see that the three most populars have been listed right here. You're going to notice they're very similar to the one you just saw because it's the same concept. So for instance, we can circulate the external media through the entire body. That is what we see with our filter feeders and the bulk movement that we experience in sponges, cnidarians, as well as in insects with their tracheal system. We can also see that you can diffuse the gas across the body surface and eventually allow the gas to mix together with your circulatory transport. We saw that, for instance, with the frog and cutaneous respiration. In fact, cutaneous respiration refers to the fact that the skin is relatively thin and moist, and gas exchange can occur through it as well. The problem with having the skin thin and moist is that these animals are often confined to some type of water environment because of the requirements of the skin, and also because it's thin, it also leaves the skin relatively vulnerable to any type of predators that might be on the look for a delicious meal. So either way, we see that the cutaneous respiration will apply to most of the aquatic invertebrates, some of the amphibians. Birds, actually their eggs also have cutaneous respiration, and obviously animal models like frogs or salamander. They will all be able to do gas exchange across the skin, or aka cutaneous respiration. And then our third option, and this is going to be a little bit higher level of organization, is that if you want to maximize gas exchange, you can utilize specialized respiratory surfaces or organs. So this would be the examples that we would see, for instance, in the specialization of the lungs and the gills in the fishes. The difference is going to be how the folding occurs. As you can see on your PowerPoint, the gills have evagination, meaning that oftentimes it forms outer pockets that extend outwards, whereas the lungs are more invaginations, meaning that they fold internally. And part of what these folds or vaginations will do is that they will allow you to increase your surface area so you can have a more effective and a quicker amount of gas exchange occur with each inhale in and exhale out. The other thing to discuss is your actual types of ventilation. So your inhaling and your exhaling pattern. How exactly does the gas come in and how does it go out? And what we see happening is that animals will often have the ability to alter their rate of ventilation, but they tend to stick to the same type of ventilation. And the three most common are going to be non-directional, tidal, and unidirectional. Non-directional means that the media that pertains to the gases comes from an unpredictable pattern. So imagine, for instance, that you're looking at a fish and it's swimming in an irregular pattern, just side to side, making loops, never knowing if it's going to go left or right. It's just free line swimming. Well, what's happening is as it's swimming, the water is interacting with the gills, and the gills don't really know, as it's waving, where exactly the water is coming from. So that would be an example of a non-directional ventilation. You don't have one entry point and one exit point. It's coming from an unpredictable pattern. Most organisms will fall either under the tidal or unidirectional flow. Tidal means that you have one area or one chamber where the media will move in and out. That's what humans do. So when you inhale, the air either enters through your nasal cavity, or for some of us, we like to breathe through our mouth, through our oral cavity, goes down the trachea into the lungs, and when it's time to exhale, we see the air basically reverses, and now from the lungs to the trachea and out to the nasal cavity, maybe some of us also our oral cavity. But it is the same opening, it is the same organ, it is the same pathway that you're moving in and out. If you see that you move in one chamber, but you exit at another one, then that's going to be unidirectional. In fact, speaking of fish, if we take a look, for instance, at sharks, what we see happening is as they are swimming around, they tend to open their mouth and the water will enter through the mouth or the little spiracles, and it will exit through the gills. And when you do that, you have one entry point and one exit point, so that will be unidirectional. As we look at our different animal models, I will try my best to definitely highlight, if we're taking a look at non-directional, tidal, and unidirectional, to sort of make it clear as to what the differences are of these methods of airflow. Another thing that I want to mention to you, because hopefully you'll see the trend line, is that there will be a combination of water breathers and air breathers when we start comparing our animal models for the respiratory system. And the reason we do this is because obviously both of them are present, and we do know that there is a concentration of gases in both mediums. However, it turns out that because water is more dense and more viscous, thicker than air, it turns out that it's going to be a lot more difficult for gases to be extracted out of water than it is out of an air medium. In fact, lots of studies have shown that the concentration of oxygen in the air is about 30 times greater than that of water. So that means that if you're comparing two animal models, one in water and one in air, the one in water is going to have to work 30 times harder to get the same amount of concentration of oxygen than the one that's an air breather. This often means that when you take a look at the ventilation methods or strategies, you're going to notice that there are two unique ones, whether you're doing a water breather or an air breather, just to accommodate the difference in oxygen concentration. So for instance, if we're doing our water breathers, a lot of them will tend to favor unidirectional flow. Remember, unidirectional means that it enters in one chamber and it exits through the other. And the reason it likes to do that is because it allows to have effective movement of the gases without maximizing the fact that the water is very viscous. So here's my little fish. And as I said to you before, if you see it enters through the mouth and then it exits through the gills, then that would be an example of unidirectional. The nice thing about unidirectional is also that what we see happening is as the gases enter through the body, it will often rely on what we call counter-current exchange. Counter-current exchange means that the flow of blood will be the opposite of the flow of the gases as it's being exposed to it. Because remember, the blood is what we're going to be utilizing as a transport media. So as the gases are entering into the organism's body, we see that when it makes contact with the circulatory system, the blood is moving in the opposite direction, allowing you to maximize how many oxygen molecules are removed from the medium and inserted into the cardiovascular or circulatory system, I should say. On the other hand, if you have an air breather, because it's so easy to extract your oxygen out of the air, we don't need to do unidirectional ventilation. We can just go ahead and do tidal, which means that it's entering and exiting towards the same opening or chamber. So for us, as I was mentioning before, our nasal cavity with our trachea and the lungs just going back and forth. We will also see that certain insects, as well as some spiders, will utilize a tracheal system. This tracheal system will be specialized in the fact that it will have a tubing setup that is able to interact directly with the air in the atmosphere and the hemolymph fluid that's floating around within the internal body of the organism. Alrighty, so now that we've kind of laid the basics, let's go ahead and take a look at some of our models of different organisms. So I think we should start off with the sponges and the Cnidarians. And I know that some of you are glancing at the PowerPoint and you're going, this PowerPoint looks very familiar. And that's because it's literally a copy and paste from when we discussed our previous chapter on the cardiovascular system. What I mean in this sense is the fact that they really don't have a respiratory system. The sponges and the Cnidarian have a very simplistic setup. They are still going to be relying on their bulk flow, very similar to when we did our circulatory system. With the respiratory system, they'll utilize the same exact mechanism. So for instance, if you take a look at your sponges, you're going to notice as water can enter through the different openings in the ostia, and as the water is exposed to the spongiculum, the little cavity on the inside, it interacts with these ciliated cells called choanocytes. And the cilia on the choanocytes are able to extract oxygen as well as any type of nutrients or gases that are required before the water will exit through the osculum, which is the larger opening on top. We see a very similar setup with our little Hydra model and our Cnidarian. The difference here is though is that they will tend to use more of a muscle contraction in the Cnidarians, allowing the tentacles to interact with the environment to create a larger surface area. And then notice how the water enters and exits through the same opening. See right here? So that would be an example of a tidal movement if we were going to classify it. On the other hand, for the sponge, since the water enters into one hole and exits through the other, what type of ventilation pattern would that be? Hopefully we all went with unidirectional because we came in one hole and we went out the other. Very nicely done. How about if we take a look at the mollusks? So if we start taking a look at animal models like our little snail right there, our clam, or our octopus and our squid. Well, what we see happening is that in this category, we can have two different mechanisms of ventilating the gills. It's all going to rely on the fact if you're going to use the cilia or if you're going to use muscular contractions. So let me show you what I mean with this. If you take a look at picture A and B, you see a representative of the aquatic snail and the clam, and notice how on both of the illustrations they went ahead and they drew the gills for you. So that's going to be your specialized organ where you're going to have your gas exchange. Now in both scenarios, these gills will be ciliated, and that's going to allow the water to interact with it as it's moving across and it's extracting the oxygen out of it. And at the same time, what we see happening is that as the water comes in one part, it exits a second chamber, right? It doesn't come in and out of the same area, which means it falls under unidirectional flow. And we can take it a step further and take a look at how the blood is moving in comparison to the media. And we see that it is a countercurrent movement, meaning that they're moving in opposite direction, allowing the maximum amount of gas to be extracted and put into the circulatory system. If you now look at more advanced level organisms within this category, like our squid or even our octopus, we are not going to be utilizing ciliated gills because they're not there. There's no cilia on the gills. Instead, what you're going to do is you're going to rely on muscular contraction. And the muscular contractions are going to be able to pump water into the cavity across the gill and then out to a secondary site. So since it's doing it, it's also unidirectional. The flow is also countercurrent. And what we see happening is because of the setup with the muscular contraction, that is then why when you look at species like octopuses and squids, that they're able to do the whole jet propulsion, where they rapidly extract water as they move at a fast pace, and as the water pumps in and out, they can go ahead and utilize it for gas exchange as well. For the crustaceans, we can take a look at, let's see, shrimp, barnacles, copepods, and what we see happening here is we are going to start off with our filter feeders, the barnacles, as well as some of the smaller species. They do not have gills, which means their only option will rely on diffusion. So interacting with the environment to get the gas to move down the concentration gradient. Shrimps, crabs, and lobsters, they will have gills that are often derived from modified appendages. And what we see happening here is that to assist with the movement, they will often utilize this little beading gill bailer. And as you can see on your illustration right there, I've highlighted it for you in the red box. And what's going to happen with the bailer is that it basically forms this beading pattern, and it's going to create a suction method to allow the water to quickly pass through the gills and exit out through little openings by the mouth. So the movement of the gallbladder propels the water out of the brachial chamber, and that's because it creates a little negative pressure. So as you can follow the black arrows, it allows the medium to enter past the gills and then exit through an opening very close to the mouth. So this would then be an example of unidirectional flow. With the echinoderms, like when you take a look at your sea stars and your sea urchins, there's also different methods for us to do our respiratory system. So in this case, the emphasis will all be on their little tube feet. So go ahead and take a look at the illustration, and you're going to notice that on the tubes, they went ahead and they highlighted some important structure. The first one is called a madreporite. This is going to be a large opening that you find on the surface of either like your sea star or your sea urchin. And this opening will be the method that water is able to enter and exit into the body of the echinoderm. And as it's doing so, it's creating this little hydraulic mechanism that can be utilized to pump through the tube feet and allow for movement to occur of the sea star. So it will kind of ventilate, go in and out of the tube feed. Now, on the outside of the tube feed, we're going to notice a few things. The first thing you're going to notice is that they have little tube feet, which almost act as a little sticking mechanism. It will allow them to anchor to the location that they're currently in. And also what we see happening is that they have external respiratory papilla. And these external papillas, as the name indicates, are where you're going to be able to extract oxygen gases from the water environment that they are in. And they're aided by cilia, which will help move across the surface, thereby expanding the surface area. Now, what about other organisms that fall in this category, besides your sea stars and your sea urchins, right? What about brittle stars and sea cucumbers? Well, they are actually going to be utilizing, instead of an external gill, they'll use an internal vagination. So let's switch over to the slide and see what's happening over there. Okay, so here is a representation of a sea cucumber. And I know the brittle star is not pictured, but both of them will use internal vagination. And that basically means that if you were to zoom into the body, there would be some section where there's massive coiling to increase the surface area to allow for better interaction with the media and the circulatory system. Now, over here on the sea cucumber, what you're going to notice is that it has what we call a respiratory tree. And the respiratory tree, as you can see, is highly folded. It has an elaborate invagination respiratory sac, and the air will get pumped in and out of the anus. And as the air gets pumped into the cavity, it is able to do its gas exchange. And as this is happening, obviously, because we're utilizing the same opening, that would be an example of tidal movement. Now, the brittle star is not pictured on this, but what we see happening is that the way its respiratory strategy will work is that it will allow water to move into its what they call bursae, which is a little, like a little cavity that it has on the inside, and it will often utilize cilia to maximize the movement of the medium. All right, so what about jawless fish? How do they go about extracting air from the environment? Keep in mind that they won't utilize the lung, obviously. Since we're looking at fish, we're going to take a look at the gills that they contribute towards the system. So let's start off with the hagfish, and then we're going to talk about the lamprey, because the lamprey is a little bit different. Now, the hagfish, and here's a little illustration of it. What we see happening is that to assist with airflow, it's going to rely on a phylum, which is a little muscular pump. You can see it right over here. And when it comes to directional flow, we're going to see that water will enter into the mouth or to the dorsals. It will go ahead and pass the phylum, and the phylum will keep pumping to generate a pressure so that the air can keep flowing internally. And when it gets to the section, it will pass through the gill sacs, allowing oxygen and carbon dioxide exchange to occur. And at the end of the day, all of the air is then filtered out through a secondary gill opening. So this would be an example of a unidirectional flow. The blood is still countercurrent, so we're maximizing how much oxygen and carbon dioxide can be derived from it. So this is the mechanism of the hagfish. The lamprey is going to be a little bit different because we have to account for it if it's eating or not. All right, so here's your little lamprey, and like I said to you, two different methods depend on if it's feeding or not. So when it's not feeding, what we see happening is its ventilation pattern is extremely similar to that of the hagfish, in that as it's swimming in its environment, water will flow in through the mouth or any type of spherical opening. It will go ahead and pass through the gill pouches and will exit the body that way. So it comes into the mouth and it exits through the gill pouches. So it would be an example of your unidirectional flow as well. So, okay, so what does that then mean when the fish is feeding? Well, when the fish is feeding, here you see him, he attaches directly to its prey, and using its very sharp teeth, it grinds an opening onto the side of the organism that it's attached to, and it starts liquefying the insides, the organs, and literally sucking it up in a parasitic method. When it's busy feeding, it obviously doesn't have time to allow air to move through the mouth in and out. So instead what we see happening is when they're feeding us, the direction of ventilation is going to change over to tidal ventilation. Tidal ventilation basically means that the water is going to go in and out of the same gill opening. And as the water is going in and out of the gill opening, It is obviously still communicating with its internal respiratory demands. It's just how you went about getting the air to diffuse across the medium. All right, how about elasmobranchs? So when we take a look at things like sharks, skates, or rays, how exactly are they going about with their respiratory system? And it turns out that they are going to utilize gills as well, but they will also utilize their buccal cavity, their mouth cavity, as well as their little spiracles, which are these like nostril-like holes that allow water to flood into their buccal cavity. And once the buccal cavity is filled, we're going to go ahead and create a pressure difference that the air can then rely on to glide across the gills, as it's not only dropping off its gases, but it's also getting ready to exit the organism at a secondary site. So let's go ahead and take a look at the little picture inserted. And as you can see, air is flowing in through the mouth and to the little spiracles. And when the buccal cavity is full, then what's going to end up happening is that it will close. And when it closes, it goes ahead and it contracts ever so lightly so that your spiracles, it will contract ever so lightly so that the pressure starts to build up into the buccal cavity. And at a certain point, the pressure is so high that the water, along with any gases that it's carrying in the medium, will be passed along through the gills. And that's it. So it comes in one way, it goes in the other way, and it utilizes its buccal cavity to create a pressure difference to make sure that you have proper gas exchange, and also obviously that it can occur in a unidirectional flow, in one opening, out the other. The story of respiration that we just had with the sharks is going to be very similar and also slightly different when you compare it to the next category, which is going to be your teleost fish. And this is because they will have what we call an opercular cavity that is covered by the operculum. And I'll show you a picture of that in 1 second. But because they have this particular flap-like extension on the side, what we see happening is that we can utilize it in conjunction with the buccal cavity to allow water to flow in and build a pressure gradient that we can utilize to flush the water against the gill, thereby extracting a lot of the gases that the fish will require for their day-to-day requirement. You're also going to notice in the step that there is a valve, an opercular valve, that will help regulate how much water flows in and out of the buccal cavity. Now, all of this might mean nothing to you right now, and that's because I will show it to you in an illustrated form so we can actually see what an operculum is and the cavity that it's going to pertain. In fact, I have a little picture that's going to come in. My apologies for the delay. Here's the picture that I was referencing to. So as you can see right here, using a little probe, you can see the flap that's being held up from the fish. That is the operculum, and that is basically shielding the little cavity that's behind it. So what I want to do now is I want to go ahead and see how are we going to explain air coming in through the buccal cavity and then being passed out by the gills that are on the side right here in their unidirectional flow. Okay, so go ahead and take a look at the illustration that's at the bottom right over here. And what it's going to tell you is that with our teleost fish, as they're swimming, oftentimes their mouth is open, allowing water to freely enter into the buccal cavity. Now the water can't go anywhere because look at the little operculum valve right over here. They are closed. So the water has nowhere to go but sit inside the buccal cavity. And as we're seeing more and more water added, the buccal cavity starts to expand. At a certain point, the pressure is just right that we can move on to the second step, which says that the mouth will close off. And at this point, the operculum valve is still closed. And we see that the buccal cavity will start compressing, generating a higher and higher pressure point. At one point or the other, the pressure is high enough that we can open our operculum valves, and we see that the medium, the water, can freely pass over the little gill arches And as it's doing so, we're obviously extracting our gas requirements. And towards the end, we do see that the mouth will open up again, allowing additional water to flow in so that the cycle can start all over again. When the mouth opens, there is a little bit of backflow of water that kind of gets sucked back to the mouth from the valve opening, but it's very minimum. But usually that's what accounts for it. Now, if you see the term ram ventilation, that usually indicates that you're looking at an organism that is utilizing respiration by keeping both the mouth and the valve open. So water can literally flow in, go through the gill pouches, and flow out of the operculum valve. Obviously, if the buccal cavity closes, then you're able to generate a pressure difference that can be utilized to cause the medium to cross over the gill pouches when the valve eventually does open. Now, when you take a look at your spiders and your scorpions in the Chelicerates category, we see that their mechanism of respiratory will be to utilize what we call book lungs. Book lungs are basically just a series of small, thin plates that are stacked on top of each other. They do create a little cavity that can be utilized to store some of the air to make it more effective for your gas exchange. In this category, we would also come across the fact that some spiders have a tracheal system. The tracheal system means that instead of utilizing your book lungs, you would have more of a respiratory system set up with a windpipe, aka a trachea, that's able to take the oxygen from the external environment and move it directly into the hemolymph or contact with the actual tissues inside of the organism. In fact, if we want to explore the tracheal systems a little bit, our best bet is to take a look at how the insects have set it up, because they have the most extensive tracheal system. So here's our little insect category, and as you remember, they have a little tracheal system, and the reason the tracheal system is so important is because, as I had mentioned to you in the circulatory system chapter, that when it comes to insects, they do not use the circulatory system as a mechanism to do their gas exchange. And the reason for that is because they have a separate system, the tracheal system, that will do that for them. The way it's going to work is that they are hooked up to spiracles, which are openings that you find on the outside of the insect's body. Most of the time it's towards the thoracic region, but either way they can be distributed all over, and the opening of the little spiracle will allow air to flow into it, and as it's flowing into it, it passes through the trachea, which humans have a very modern form of that. It's your windpipe. The trachea will start subdividing into tracheoles, so smaller pieces, until it's comfortable interacting with the actual muscle that it's trying to supply with oxygen and pick up its carbon dioxide. Usually what we see is that the ends of the tracheals are usually filled with hemolymph, making it a little bit easier for the gas exchange to occur. So the gas will diffuse in and out. And like I said to you before, this is one of the main reasons why when you take a look at your insects, it's always going to say something along the fact that insects do not need to have a completely separate ventricle, and that is because they're not utilizing their circulatory system for their gas exchange. The other thing that you want to think of is to actually keep the air flowing. We see that they can use the contractions of their abdominal muscles as well as the thoracic, so like when they're flapping their wings, it can change the size, and then therefore it can change the gradient of the structure that you're looking at. In that case, very similar to when we talked about the fish, we would have to study the direct patterns to figure out if we're looking at a tidal or a unidirectional type flow. Both of them which are possible, because if you're doing a tidal ventilation, that just means that you use the same spiracle to enter and exit. Whereas if you're doing unidirectional, then maybe you have spiracles towards the front of the body, and as the air is exchanged or the gas is exchanged with the body, they will exit through a more of a abdominal or dorsal spiracle, one that's more down the line of the body. And also, because of the flight pattern that a lot of insects have, they are able to do ram ventilation, which is the same thing that we saw with the fish, meaning that as they're inhaling and exhaling, they're actually doing this with their mouth open, allowing the air to drag along the body and therefore extracting the gases as are required. And a pretty cool thing about their trachea is the fact that it is able to do expansion and contraction, and in some animal models it can actually increase its size by 50%. So imagine how much control you'll have over the amount of air that's actually entering and exiting the body. That tracheal system of the insects is a very unique and a very interesting structure to kind of explore when it comes to the circulatory as well as the respiratory systems. All right, if it's okay with you, I'm going to have this one be a little shorter. So it's not a full hour. It's going to be a little bit under 40 minutes. So this chapter isn't as long as the others, so that's okay. So we'll take a break over here. And as always, questions, comments, concerns, feel free to reach out to me. And in a little bit, hopefully I'll see you guys for part 2. And we'll continue to look at different animal models and their respiratory methods. Thank you.